Hi, today I'll be talking about the process of protein production in bacteria. Um, I'll mainly go through the bacterial transformation, the protein production, and the extraction process. Here's a timeline, tentatively the number of days it takes between doing the bacterial transformation to checking the protein purification using STS page. So the first thing is um, the E. coli transformation using the T7 Express strain, which is widely used for protein production process. Before you get started with the experiment, make sure you locate your plasmid, which should be in the -20, and you should also be able to locate the T7 Express cells, which should be in -80. And then once you're done with that, start preparing a water bath at 37 degrees centigrade and also prepare wet ice. Um, Please make sure that the water bath is ready before you get well into the process, because once you start incubating and then you realize that the water bath is not ready, it just not only delays the process, but also can affect the transformation efficiency. So while these are done, then you can start by thawing the T7 Express cells on wet ice, and then you pipette the plasmid directly into the cell, and the volume will vary based on the concentration and the transformation efficiency. Then you can tap uh, gently to mix it. I prefer pipetting it very slowly to do a homogeneous mixture, but make sure it's not too vigorous. And then once that is done, incubate it on wet ice for about 30 minutes. While you're incubating it, store the extra plasmid at -20, and also make sure that you have SOB, which you're going to use in step 8, in liquid form. Usually it should be in the refrigerator. If it's not, then it's definitely with the T7 express cells in -80. So make sure you take that out and thaw it and bring it to room temperature before this incubation step number 4 is done. So after this, you take your cells and place them again on the wet ice and take it to the water bath. Make sure your wet ice is near the water bath. Because you'd be heat shocking it for exactly 90 seconds in 37-degree water bath, and then you'd be placing it into the wet ice for 5 minutes. Timing is the key. Also, as soon as you're done with the 5 minutes incubation, then you take the SOB and add 950 microliters of it into the cells directly. Once that is added, you take the vial and put it on a shaker at 37 degrees centigrade and shake it for 1 to 2 hours at 250 RPM. And also make sure that it's at an angle for better circulation. While you're incubating this or shaking this, you can start preparing the LP agar plate with a proper antibiotic if it's already not prepared. Make sure you add your antibiotic after the LP agar cools down to about 55 to 60 degrees centigrade. If it's too hot, then the antibiotic might not work. If it's too cold, then the LP agar starts solidifying and the antibiotic would not spread evenly and it would not help in the transformation. And then once it's done, spread about 200 microliters of the transformed cells on the agar plate. 200 microliters is kind of the optimum volume that you can put. If you put too high volume of the cells on the plate, it might be overcrowded. But based on how the transformation efficiency is, you can ballpark around 200 to 300 microliters on the agar plate. And then you can incubate the inverted plate overnight at the 37 degrees centigrade incubator. And the remaining transformation can go back at 4 degrees centigrade, but also make sure if you're unable to transform or plate everything, make sure you plate the rest of the cells within 2 to 4 days, because the transformation efficiency drops significantly, and you would probably not get good colonies, or you'd probably get dead cells, or not great transformation if you keep it longer. And hopefully, the next day when you come back, you see beautiful separate colonies, and then you can start with the protein production. Um, so for that, you need 5 mils of LB, and you add your antibiotic into the test tube, and then you can transfer a single colony of the E. coli cell and then let it shake in the incubator at 37 degrees centigrade, 250 RPM for overnight. Not more than 36 hours, though, because you do want to catch it at its saturation phase where there are not a lot of dead cells. So once that is done, you now transform it or you transfer it to a bigger flask. So you take a 2 liter flask and add 1 liter of LB in it. Do not make the mistake of adding more LB that I did in the initial days when I was trying out this process. I got greedy and I thought, okay, I'll add a little more LB to get a little more protein yield, but that doesn't really help because if you put more than 1 liter in a 2 liter flask, while it's shaking, the chance of it getting contaminated or spilling um, is enormous and it's not worth the risk. So stick to 1 liter in a 2 liter flask. So once once you're done with autoclaving the new LB and putting it in the 2 liter flask, then you add the saturated culture into the flask. So before you add this, it's also important to check the OD of the starting culture because that would help you to decide how many hours or how many minutes you're going to put um, the incubation on for this larger flask. So you want the final concentration or the final OD of the larger flask to be between 0.5 to 1 because if it's more than 1.5 or 1.6 um, as it approaches 2 basically, then the protein yields will drop drastically. So you would want to catch the cells when it's at an OD of near about 1. So make sure you're keeping a track of time and also checking the OD every 20-25 minutes or so. And again, if you're unsure of the pattern of how it's growing or how the protein production is happening, it's always advisable to take out 50 mils of the culture, spin it down, take the LB out, and then resuspend it in water, and then do the SDS pH comparison. So once the larger flask or the larger culture reaches an OD of near about 1, then you can start the induction process. And for that, you would be adding 1 milliliter of 0.5 molar IPTG such that the final IPTG concentration is 0.5 millimolar. And for induction process, you would want to reduce the heat. So so far it was shaking at 37 degrees centigrade. Now you would want to keep the temperature below 20 and you may open the door of the shaker for about 5-10 minutes to let it cool. I prefer turning the fan off and sometimes even switching it off and then opening the door to let it cool even faster. And then once it's done, like once 10-15 minutes goes by, then you can restart the shaker again and then let it and set the temperature below 20 so that it slowly comes to room temperature, and then you can set it back to room temperature and let it run for how many ever minutes or hours that you need to induce it for. Here it's recommended to do it for 1 hour, but it's important that you find out the optimal incubation time because it would be varying from protein to protein. And the way to do is obviously taking out several timestamps and then checking it using SDS page. This is something that I did when I was initially trying out the different incubation conditions for my protein. Here is the SDS page with the ladder in the first column, and then the 2 and 3 are the 2 different types of proteins that I was using. It was prior to inducing it with the IPTG, and then inducing it for 45 minutes, 90 minutes, 360, 150. And clearly the 90 minutes signal was the highest. So for my proteins of interest, the 90 minutes incubation seemed to be the most efficient one, and I stuck to it. Sometimes if I ran out of time or I didn't have enough time to continue the process, I would let it run overnight. The protein production would still be okay, but it would not be as good as how it would be when it's a 90 minutes induction. Um, so once that is done, the next process is doing the lysate, which I personally really like doing it. And it's very satisfying in a way, because you get to squeeze out the good stuff, and then uh, you're closer to your protein in soluble form. For this, you pellet the cells. So once you've collected the induced uh, culture, you pellet the cells for about 15 minutes. Take the supernatant out and then resuspend it in the lysis buffer. So this is the recipe of the lysis buffer. Also, make sure you add the protease inhibitor so that you get better yield. Because in some of the protocols, they don't mention the protease inhibitor, but I always found it useful to add it to my lysis buffer. So once that is done, resuspend the cell pellet in the lysis buffer, and you can transfer it into a 15 ml conical. If you do not have time to do all this on one single day, the first step is where you can stop, where you can. So after you've removed the supernatant, do not add anything and just freeze it, and just put it in -20 if the pause is needed. If not, then you can continue with the rest of the process. So once you have added the lysis buffer with the protease inhibitor and resuspended the cell pellet, then you take it and transfer it into a 15 ml conical. So there are a few different ways you can do the cell lysis. One option, which is quick and easy, is the freeze thaw, which is also super efficient for some of the proteins, where you defer the temperature so much that you help the cell walls to burst out. And then once you have done the freeze thaw method for about 4 or 5 times, then you centrifuge it, take the supernatant out, and filter it, and collect the filtered product. The other form of cell lysis is using a sonication gun, which we have at IQ in the cold room. So this is also something that's super interesting and easy to use. Basically, you take your lysate in the 5 ml conical, and 15 ml conical, and then you put it inside, or you take this probing sonication gun and you put the conical in, and make sure your conical is sitting in ice bath the entire time, because you don't want your protein to denature due to excess overheating. The typical settings that I use are pulse on for 0.8 seconds and pulse off for 0.2 seconds, and use it for 6 cycles, which has 10 seconds off on and 30 seconds off. You can play around with the settings depending on trying out a few different samples and seeing which gives you the best protein yield. And then finally, once the sonication is done, it should give you like a frothy cell suspension, and then you can centrifuge it at 4,500 RPM for about an hour and collect the supernatant and filter it out. The other process of cell lysis that I personally love using is French press. As the name suggests, it obviously uses pressure as the main attribute or the main parameter for doing the cell lysis. So there's this pressure chamber, and here is the French press in silver. It is not light at all. This thing weighs about 20 pounds, so it's quite heavy. Um, so the piston, this tiny, let me zoom in. Yeah. So uh this narrow thing is a piston, and then it has like the collection valve here at the bottom. So y- before you start the process, you want to make sure that your French press is sterile, and it's also kept in the refrigerator for about 30 minutes before you start the actual process. It's highly recommended to do the assembling and putting the cells in the cold room before you transfer it and take it to the pressure chamber. So it's also important that you lubricate the O-rings of the piston and also the valve so that there is not a lot of friction, and because it's metal to metal, it can cause abrasion while it's being well, it's being um, pressurized for about 1100 psi. So this is how it looks like in an inverted position. So you have the piston that's sitting down, and you can push the piston up till there is a line that demarcates the fill line. So you can push it up as much as about a quarter of an inch, and then you would have enough space for your cells to be poured into this cavity. If you have more than 35 ml, please run your samples in different aliquots. But if you have less than 35 ml, um, you should be good to go in one run. So once you've poured in your sample, then you place this back in and make sure that it's not super tight, because then there is this chance of spilling. And also, um, this is the collection valve. The collection valve also has an outlet from which you would be collecting your actual sample. So while you're assembling everything, make sure this collection valve is kind of facing away from you, because sometimes it would just, accidentally splash, or it would just spill out. So you would also want to make sure that there is a collection tube that is attached to this collection valve is always there while you're putting this top portion, mounting it on top of the French press. And once everything is assembled, you invert it, make sure that this is secured and it's not, falling down. And then you place it here, and then you attach everything, and you can start the pressure chamber and set it at a pressure of 1100 psi, which is perfect for the E. coli cells to burst open and you know uh, get the good stuff going out. And then you would be able to collect your um, lysate in a collection tube using this collection chamber. And also make sure that it's always in wet ice so that it's not overheated and it doesn't get denatured. And then the same procedure, collected, centrifuged at 4,500 RPM for an hour, and then filter it out. Then you can store it or use FPLC directly to purify your protein. And once the purification is done, you can definitely use SDS page to confirm whether you have your protein or not. So this is in short the first three main processes that are involved in the entire protein production using bacteria. I hope this helps. If you have any questions, please let me know. I'd be happy to share more insight on the French press process. Thank you.